Welcome to part 11 of this video on the immune system. Okay, so let me just give you a quick summary of where we've got to with our discussion of the adaptive immune system. So far, we have seen that an antigen-presenting cell is going to present fragments from antigens of our pathogen on its cell surface attached to either MHC class 1 molecules or MHC class 2 molecules. And remember, if it's an intracellular pathogen, you'll have the antigen fragments put on MHC class 1 molecules. If it was extracellular, either because it's only extracellular or because at the time when the antigen presenting cell found it was extracellular, and remember we're thinking about intracellular pathogens having to go extracellular for a while, it's going to have its antigen fragments presented on MHC class 2 protein complexes. And remember, we're now going to use these antigen fragments on MHC class 1 complexes to activate the cell-mediated adaptive immune response, and these antigen fragments on MHC class 2 complexes to activate the humoral adaptive immune response. And both of those adaptive immune responses, they rely on us activating T cells, T lymphocytes, first at least, anyway. So what we now want to discuss is lymphocytes. So rather than trying to continue this on in a chronological order, which would, I think, be very difficult to do, what I want to do is take a break now and discuss lymphocytes for a long time. I want to discuss how you produce lymphocytes, how they circulate around the body, and then we'll come on to how they are activated by the antigen-presenting cells. But firstly, I want to discuss their production. And the main reason I want to do this in a huge amount of detail is that lymphocytes are all specifically directed against certain antigens, and I want to explain how that comes to be. And that's a long discussion, so we're going to do that aside from our discussion of the actual adaptive immune response process. Okay, so we're going to take a bit of time out then and discuss lymphocytes. So, lymphocytes then, they are special types of white blood cells, so lymphocytes are a subset of leukocytes. And overall, you have a huge number of lymphocytes within the body. You have 2 times 10 to the 12, it's estimated, lymphocytes. That is a very large number. That is 2 trillion lymphocytes. That is a similar cell mass to the liver. So in total, if you were to clump all of your lymphocytes in your body together, you would have a similar cell mass to the size of the liver. So you have a huge number of lymphocytes inside your body. And lymphocytes can be divided up into 2 major different types: the T cells and the B cells. And we are initially going to focus on the T cells, and we will later on talk about the B cells. And more properly, you can call them T lymphocytes and B lymphocytes, but we'll just refer to them as T cells and B cells. Okay, so what I now want to do then is focus in at the moment just on the T cells, and I want to discuss where they come from, so their production and their development. So how they actually come to be specialized against a certain fragment of a certain antigen. And then I want to discuss how they circulate around the bloodstream and the lymph nodes and the lymphatic vessels. And then finally we'll go back to the story and look at how antigen-presenting cells are going to activate T cells. Okay, so let's start off by discussing the production then of T lymphocytes. Now, this is a complicated story, and the major complicating factor is that the thymus gland is extremely important in the production of T cells. But the thymus gland doesn't stay with you for your entire body. The thymus gland gradually involutes away, which means it gradually just disappears and is replaced by fatty tissue. So production of new T cells is believed to just occur in childhood and early adulthood, but then by later adulthood, it's not really understood particularly well where the new T lymphocytes come from, but I will explain to you what the major theory, the major viewpoint is as to how you maintain your population of T cells. So initially we're going to talk about the production of brand new T cells, however, and therefore we're going to assume that we are currently working in a child's body. So let's assume we are currently a child and we're going to follow the pathway for producing new T cells. And we'll talk later about what happens in adults once the thymus has gone. Okay, right, so let me go through the pathway then for producing new T cells. So the story begins with cells within the bone marrow, specifically the hematopoietic stem cells. So let me just draw a picture of this. So we'll have a picture of the femur here, and I'll show the bone marrow inside the femur. So these, starting distally, these are the medial condyle and the lateral condyle of the femur. Here's the shaft of the femur, the greater trochanter here, the neck of the femur, like so, and then the head of the femur here. Okay, so there's my picture of the femur, one of the long bones, which will contain some bone marrow, and the bone marrow will be in this sort of region. So of course, the outer layer of the femur's shaft will be very solid cortical bone, but then inside you'll have a hollowing, and this will be where the bone marrow will be placed. Okay, so in red here, this is the place where all of the bone marrow is present in the femur. Okay, now in the bone marrow you have a special type of cell known as a hematopoietic stem cell, and these cells are responsible for the production of red blood cells, white blood cells, and also platelets. So it's responsible for all of the different cell types that are within the bloodstream, hence its name, hematopoietic. So you're going to have within the bone marrow a special cell type known as the hematopoietic stem cell. So let me now just draw this hematopoietic stem cell out. So here this can represent our hematopoietic stem cell, and it will be present within the bone marrow. So I'll just colour him in red here. Okay, so what's going to happen then in our production of T cells is the hematopoietic stem cell, it's going to divide and divide and divide and divide, and some of its progeny will remain as hematopoietic stem cells to maintain the population of hematopoietic stem cells. Other of its progeny will turn into new cell types, and one of the cell types that it's going to produce is known as a lymphoid progenitor cell, and this is the cell from which T cells are going to be made, and in fact also B cells are going to be made. So here you're going to produce lymphoid progenitor cells. So this is a lymphoid progenitor cell. And these lymphoid progenitor cells, they can either become B cells or T cells. Now, if they're going to become B cells, they're going to remain within the bone marrow, because the bone marrow is the site of B cell development. If they're going to become T cells, they're going to leave the bone marrow and they're going to go up to the thymus gland, because the thymus gland is the site of T cell development. So it's quite convenient. B cells develop in the bone marrow, B for bone marrow, and the T cells develop in the thymus, T for thymus. Okay, so the hematopoietic stem cells then, as I say, they're going to divide and divide and divide. Some of them will remain as hematopoietic stem cells, some of the offspring will become lymphoid progenitor cells, and these lymphoid progenitor cells are now going to leave the bone marrow and they're going to go up to the thymus. So temporarily, of course, they'll have to go into the bloodstream to get to the thymus. So I'll just, to complete the picture, we'll have the bloodstream shown here, so they'll go into the bloodstream. And then the bloodstream is going to deliver them up to the thymus gland. So let me now draw you a picture of the thymus gland. And again, I'll stress that this is a gland that you can only really find in children and young adults. And it will be gradually getting smaller and smaller in the young adults. And it just gradually disappears and is replaced by fat. And therefore, this production line will stop in adults, and something else is going to have to keep our supplies of T cells up, and I'll discuss the major viewpoint of what that is later on. Okay, so let me draw a picture then of what a thymus gland looks like in a child. So a thymus gland, it looks like a butterfly. So here is my picture of the thymus gland. It's similar to the thyroid gland, but with a much thicker connection between the two sides. So here then is my picture of the thymus gland. So this is representing the thymus gland, and the thymus gland, it sits in between the two lungs. It's in the mediastinum, the anterior mediastinum, just behind the sternum, in between the two lungs and in front of the trachea and the carina. I'll draw a little picture of that in a moment. So I'll colour it in here in green, and this pen's going, but never mind. Right, so there's my thymus gland, And now let me draw a picture of where it would actually be positioned with the lungs. So I'll draw the lungs firstly. So here, this can represent our right lung, and then let's have the left lung here, like so. And then of course this is the cardiac notch where the heart would be, but I won't put in the heart. Here is the trachea, and it bifurcates with the carina here into the two major bronchi, the right and left bronchi. And now the thymus gland is going to be positioned in front of the trachea at this sort of position, like so. So in green there, and of course it will just be behind the sternum, so if you're thinking about the chest wall, the sternum will be anteriorly and it will just be behind there. Okay, so there's the thymus gland. So these lymphoid progenitor cells are going to come to the thymus gland. What I now want to discuss is what's going to happen to them in the thymus gland, because of course in the thymus gland they are going to become T cells, and very important processes are going to occur to make these T cells specific to a certain antigen fragment. So let me now, oh, I need another piece of paper, just bear with me whilst I get another piece of paper. Okay, right. So what I now want to draw is a more in-depth picture of the thymus gland. So I want to go onto the histological scale and have a look at what the thymus gland actually looks like if you take a little portion of it and zoom in and in and in. And I want to have a look at the different areas of the thymus gland where the lymphoid progenitor cells are going to go and what's going to happen at different stages to turn them into T cells. Okay, right, so let me now draw a picture then of the thymus zoomed in. So if I just bring back up my big picture of the thymus. So what I'm now going to do is just take a little section of it here, so this little box here, and I'm going to zoom in and in and in on there, and we're going to have a look at the microanatomy of the thymus gland, and we'll see the different areas that are going to be involved in T cell development. So firstly, the outermost layer of the thymus gland consists of a capsule, so that's the layer that we're going to have to start off with. So this layer here, and I will just colour it in green, this is going to represent the capsule of the thymus gland. So this is our capsule. Now there are extensions of the capsule into the tissue of the thymus gland, and this actually is going to split the thymus gland up into different lobules, as they're called. So I'll just draw a few of these on here. So here is an extension of the capsule into the tissue of the thymus gland, and it, as you can see, is going to divide the thymus gland up. So I have another one of them here into separate little areas which will be known as lobules of the thymus gland. So I'll also colour these extensions inwards in green here. And these extensions into the sort of tissue of the thymus gland of the capsule are known as interlobular septums. So they're septums between lobules. So this here, this is an interlobular septum. Okay, right. So now let's actually have a look at what's inside of the capsule and between the interlobular septums then. So we divide the tissue of the thymus gland into different regions depending on how close it is to the centre. So, and these different regions are important because the lymphoid progenitor cells are going to go to the different levels at different stages of their development into T cells. Okay, so the region of the thymus tissue that is just underneath the capsule here, this is known as the subcapsular region. So we'll start off with this region here. So this is called the subcapsular region. Then underneath the subcapsular region you're going to have what's known as the cortex of the thymus gland. So again, I'll put dashed lines to show this. So this portion underneath here, this is now known as the cortex of the thymus gland. Underneath the cortex, you're going to have a region known as the corticomedullary junction, and then the most central portion of the thymus gland tissue is then known as the medulla of the thymus gland. So again, I'll just put another dashed line here. This innermost portion here, this is now called the medulla in here, and then this section in between the cortex and the medulla, this is called the corticomedullary junction. Okay, right, so these are all the different regions that we divide the thymus gland tissue up into, and it will be relevant because we'll see that at different stages of development the lymphoid progenitor cells and the developing T cells are going to go to these different areas, so it will all be useful. Right, so what I now want to talk about is how are the lymphoid progenitor cells actually going to gain access into the thymus gland. So in the corticomedullary junction, there are special types of venules known as high endothelial venules, and we're going to see high endothelial venules again later in lymph nodes. The reason they're called high endothelial venules is that they have a very high endothelium. So if I just draw a high endothelial venule here, so the endothelial cells of blood vessels are usually extremely flat. They're usually squamous. They're not usually cuboidal in shape. In these special high endothelial venules, which we find in the corticomedullary junction of the thymus gland and also in the lymph nodes, as we'll see later on, the endothelial cells are much, much taller, and therefore these venules are known as high endothelial venules, and they're involved in bringing the lymphoid progenitor cells out of the bloodstream and into the thymus gland, and later they'll be involved in taking the T cells that have developed in the thymus gland back into the bloodstream from the thymus gland. So this is what's known as a high endothelial venule, and these high endothelial venules are present in the corticomedullary junction region of the thymus gland. So this is a high endothelial venule. So remember, a venule is a blood vessel after the capillaries have reconverged, as we've discussed previously when we were discussing the acute inflammatory response. Okay, so here is our high endothelial venule, and I'll just give it a basement membrane for the endothelial cells to sit on. So the endothelial cells are very tall, hence why they're called high endothelial venules, and their special function is that they're capable of bringing the lymphoid progenitor cells out of the bloodstream and into the thymus gland. And once these lymphoid progenitor cells have been brought into the thymus gland, they will move from the corticomedullary junction up to the subcapsular region up here. So I'll draw one of these lymphoid progenitor cells up here, and I'll try and keep the colour coding consistent, so we'll have it in orange here. And it's going to move up to the subcapsular region from the corticomedullary junction where it was brought into the thymus gland by the high endothelial venules. Okay, so to summarise where we've got to so far, the hematopoietic stem cells in the bone marrow have produced lymphoid progenitor cells which have gone into the bloodstream, and they're now arriving in the thymus gland. They're coming out through these high endothelial venules, and they move from the corticomedullary junction up to the subcapsular region. What is now going to happen to these lymphoid progenitor cells to turn them into T cells? Well, they are going to develop their own unique T cell receptor, which is going to be able to recognize a certain fragment of an antigen. Okay, that's the key here. It's going to be able to bind to an MHC complex with a certain peptide fragment bound to it. And T cell receptors, they might be able to bind to MHC class 1 with a certain peptide fragment between 8 and 10 amino acids long, or it might develop a T cell receptor that can bind to an MHC class 2 with a peptide fragment between 12 and 20 amino acids long. But we are going to develop on each of these different lymphoid progenitor cells a different T cell receptor that will be capable of recognizing and binding to a different MHC peptide complex. Okay, so what I'm now going to talk about then is the structure of this T cell receptor, and then what we need to go over is how does the T cell receptor end up becoming unique on each of these different developing T cells. So firstly then, the structure of the T cell receptor, the easy bit, and then the more difficult bit of explaining how they actually end up with a unique one. So let's say that this is the cell membrane of our T cell, or indeed our developing T cell. Okay, the T cell receptor, just like MHC proteins, is going to consist of two separate polypeptides which are bound together. And these polypeptides are again known as the alpha chain and the beta chain. So immunologists use the same names over and over again. So we call the two different chains of the T cell receptor the alpha chain and the beta chain as well. And again, the alpha chain and the beta chain have two of these important domains each again. And in fact, the picture is going to look almost identical to what I drew for MHC class 2. So here, let's say this is the alpha chain. All that's different now is that these two different domains are called something different. So rather than calling this domain the alpha 1 and this domain the alpha 2, which we did in the case of MHC class 2, this is now called the variable alpha region, and this is called the constant domain of the alpha chain. Okay, and hopefully you should be able to guess which portion is going to be different between different T cells and which portion is going to be the same between different T cells. So this is the alpha chain of the T cell receptor here, and here then we'll also have the beta chain. So again, the beta chain is going to have two important domains. It's N-term, this is going to be extracellularly. It's then going to have these two important domains, the variable domain of the beta chain, and then the constant domain of the beta chain, like so. And then C-terminus will be here, and this will now be the beta chain. And I'll colour the beta chain in green again. Okay, so this is the beta chain. So this is the structure of the receptor that T cells are going to have on their surface. So we're going to produce a huge, great population of T cells, and every single one of the ones that we produce is going to have a different T cell receptor on its surface. The alpha chains and the beta chains that it uses to produce its T cell receptor is going to differ compared to other T cells, and this is going to allow it to recognize a certain MHC peptide complex that no other T cell can recognize. So this is how T cells are specifically tailored against a certain peptide fragment. Okay, so this receptor then is known as the T cell receptor, and for short, the T cell receptor is often abbreviated down to TCR. So T cell receptor. Okay, right. So the first part then in turning this lymphoid progenitor cell into a T cell is for it to develop a T cell receptor. We need it to develop its own unique T cell receptor. And what we now need to discuss is how do these T cell receptors come to be different on all the different T cells. And that process is very complicated, and it's known as VDJ recombination, and we will go through that process in the next video. But for now, just know that it is the variable portion of the alpha chains and the variable portion of the beta chain that is going to vary for the different developing T cells. So, if we were to, so let's imagine that we've got loads of lymphoid progenitor cells that have now undergone the process of creating their T cell receptors, and after they've undergone the process of creating their T cell receptors, they're no longer called lymphoid progenitor cells, they're instead called thymocytes. We'll come on to that in a moment. But if you were to look at all of their different T cell receptors, they'd all have the same constant regions of the alpha chain and the beta chain, but the variable regions of the alpha chain and the variable regions of the beta chains would differ for the different thymocytes that you have produced. So you're now going to get each of the different lymphoid progenitor cells to produce its own unique alpha chain, its own unique beta chain. It won't change this constant portion of either of them, but it will change the variable portion, and I will go over in the next video how they come to have their own unique alpha chain with its own unique variable domain, and their own unique beta chain with its own unique variable domain as well. Okay, but we'll have a break here and do that in the next video.